This presentation on propensity score matching was prepared as a part of HAP 819 course on Advanced Statistics in Health Services Research taught by Dr. Alemi at George Mason University in the Department of Health Administration and Policy. In electronic health records, we don't have randomized treatment and control group with the same distribution of covariates. Due to lack of randomization, the covariates and effect of treatment are confounded. So how will we solve this problem? With the propensity score methods, which will allow us to minimize the effects of observed confounding when estimating the treatment effects using observational data. For this assignment, we will use data from National Institute of Mental Health. Use of a specific antidepressant is our treatment. So, we have two groups under the treatment. One, group of patients who have received the specific antidepressant, and the other group where the patients did not receive our antidepressant. That means they have received an alternative option. Now we want to explore the effect of a specific antidepressant on the remission of depression. So, the remission of depression symptoms is our outcome. Patient's age, gender, and medical history can affect both the treatment selection and the outcome. And now we will have to conduct a propensity score matched analysis to create comparable risk groups. This is our assignment. Now, how will we do it? These are the steps for propensity score matching. I will go step by step. The first step is selecting and listing the covariates of our interest. It is preferable to include those variables which can affect both the treatment selection and the outcome. Now the question is why? Because we want to induce a balance on the variables that can predict both the treatment selection and the outcome. So in our case, we will select such variables which have potentials to influence both the use of antidepressant and our outcome, the remission of the depressive symptoms. And just to remind you, our main goal is to make the two treatment groups comparable with respect to all possible observed variables and confounders. If we exclude any important variables or covariates, it may add bias in our matching. This is our list. We are considering more than 30 variables as covariates before matching. Now, the question is, how many variables should we include? Well, the propensity score matching doesn't penalize us for a big number of covariates or variables as long as we have a good sample size. We are trying to make the group comparable as much as possible. Excluding any important variable may add bias in our result. Before step 2, let's do an experiment. It will be interesting. What will happen if we try to conduct our outcome analysis with our non-randomized observational data without any propensity score matching? You can see the codes here, and when we run the codes, we found no significant effect of treatment on outcome, and it seems like many variables are confounded. So we can save this result for now. When later on we will employ the propensity score methods, we can compare this result with our final one and see how controlling for confounding through the propensity score matching changed our result. Now we will create the propensity score. As we have two treatment groups, used antidepressant versus did not use our specific antidepressant, we will have a binary outcome. Then what is the most common model for a binary outcome? It's logistic regression. So we will use logistic regression to match the covariates and estimate our propensity score. Now, when we will fit our variables into the logistic regression, what will we get from our logistic regression? We all are familiar with the concept of log odds. So what goes into the odds? Probabilities. So after running our regression command, we will get the predicted probability of all the patients. And now, once we have all the probabilities, the matching starts. You can see here we got our probabilities. To be more specific, predicted probabilities 0.52, 0.78, and so on. So propensity scores are these probabilities. Based on our probabilities, now we will match the subjects that received our treatment, that is, received our specific antidepressant, with those who did not receive that antidepressant. Now you can see two patients have exact same probabilities, 0.78. Hopefully, this pair will have the comparable feature. And this is 1:1 nearest neighbor matching, and it is exact matching. In real life, it is difficult to do so. So what will we do in real life? We will try to identify the closest neighbor to make the pair. For example, red and pink pair, 0.78 versus 0.77, 0.01 difference. Usually a small difference is allowed, for example 0.0001, and this is called caliper. We start with a smaller difference as caliper to make pairs, because the smaller the difference, the better the matching. And another idea to identify the pair is inverse probability of treatment weighting, and this is our next step. Now our third step. Using the entire sample, we will create weights, 1 divided by the propensity score matching for the use of antidepressant, or 1 divided by 1 minus propensity score matching for not using the antidepressant. In this way, we will calculate all the weights for all the patients, and this weight will be used as a new variable in the dataset. Now we will assess the propensity score so that we can understand whether we are on the right track. We have identified our pairs. Say we have 50 pairs for two groups. So think of this. Are the groups comparable or not? Propensity score is a balancing score. It is conditional on the distribution of the variables. Before proceeding for outcome analysis, we will have to make sure that the participants from two groups, treatment and no treatment, have the same distribution of the measured variables or the covariates. How do we do that? We will use standardized difference to quantify the differences in means or prevalence between the two groups. For balanced diagnosis, we have developed the weighted model. After running the code, let's see the result. The weighted regression removes the effects of all covariates. Now, how do our matched pairs look like? This is the code for graphical presentation. Are the two groups comparable with respect to all possible observed variables and confounders? Pretty much. However, there is an attribute that has a statistically significant impact on the use of antidepressant even after applying the propensity scores. Now we are concluding with outcome analysis. We will carry out weighted regression of remission rates on various covariates and use of antidepressant. Here, as you can see, the dependent variable is now remission rates. The treatment variable, use of antidepressant, is now included as an independent variable. Now we will run our code and wait for the result. Do you remember the experiment we did without propensity score in the beginning? Now we will compare the result with our final outcome analysis after matching. Here you can see that there is a statistically significant relationship between the treatment and the outcome after balancing all the effect of the covariates. So this was our assignment, and now a few questions before wrapping up. Can we calculate propensity score for the missing values with treatment and outcome? It's better to drop those observations, because if we calculate a propensity score with the missing values, maybe we will get biased results. And in our assignment, we have two groups for the treatment. Can we calculate propensity score for more than two treatment groups? Yes, in that case, we will have to conduct a multinomial regression. This is the basic of propensity score matching using R. Hope this was helpful, and thank you for stopping by.